Hi everyone, and welcome to another fantastic Teva API webinar. I'm Tammy Ben David, and I'm your host for today. Wherever you are in the world, thank you for joining us, and we're very happy to have you. So today's webinar is on the topic of nitrosamines, a moving target, and we have a fabulous lineup for you today. First up is Lena Ben-Moha-Lerman, who is Senior Director of Lifecycle Management within Teva API's R&D team. Lena holds a PhD degree in organic chemistry and has worked in Teva API for 14 years. We're also pleased to be joined by Diana van Greenhoven. Diana has an MSc degree in chemical engineering. She has been at Teva API for 4 years and is currently head of global compliance at Teva API. We'll also be hearing from Vesna Pergomet. Vesna has 15 years of experience in pharma regulatory affairs and has been at Teva for 4 years now. She has a bachelor's degree in chemistry and an MBA. Moving on to the agenda for today, the evaluation requirements for nitrosamines in medicinal products has evolved over the last 3 years. For APIs, this is a really essential topic. Lena and Diana will be taking you through the changing nitrosamine regulations and showing you our critical 3-step approach: risk assessment, confirmatory testing, and change for marketing authorization. You'll learn about the history of nitrosamines impurities, principles behind the guidelines and Teva API's vision, and lastly, how to avoid nitrosamines in any and all medicinal products. We encourage you to please ask any questions you may have. There will be some time for questions at the end, so please feel free to pop any question in the bottom of the screen and we'll get to as many as we can. And our lovely speakers will answer them. So now, enough of the introduction. Let's get started. Diana, over to you.
Hi, my name is Diana van Greenhoven. I'm heading the global compliance at Teva API. Nitrosamines, a moving target. That's what we made for the subject of this webinar. There has been quite some things changing in the last 3 years. First of all, there were changes in the nitrosamines to look for. It all started with the NDMA, which was found in 2018. And nowadays, the guidelines have a lot of more nitrosamines mentioned to look for. Second point, if we go from left to right, changes in the products to include. As I said, it started with NDMA, which was found in valsartan, and it evolved to include all the sartans and later on the chemical synthesized APIs. And nowadays, we also have to look for fermentation products and also biosynthesis products. The next point is changes in the timelines to adhere to. We have had some extensions for the timelines to give information about the risks for nitrosamines. This was also mainly due to some changes due to the pandemic situation which we are in. If we then look for the changes in root causes for nitrosamines, it started all off in the route of synthesis. So the focus was for route of synthesis because to form a nitrosamine, you have to have 2 components to form the nitrosamine. And therefore, in first instance, the route of synthesis was one of the main root causes for nitrosamines. But later on, industry, as well as regulators, saw that there were other root causes for nitrosamines, specifically also cross-contamination root causes, which could give nitrosamines in products where you would not expect them to be. If we go to the next point, it's changes of acceptable intake requirements. In the beginning, specifically in Europe, there were specific acceptable intake requirements for the sartan family for an interim period of 2 years, while especially also US FDA issued other kind of guidelines for the sartans where they specifically said that it should be undetectable. Nowadays, the guidelines are more coming together and the acceptable intakes are spelled out for the different type of nitrosamines as well as the different type of products. Last but not least, changing in testing methods. A lot of testing methods for the nitrosamines have been published the last 3 years, and there were all different kinds of level of detection, level of quantification. And now in the guidelines, it's spelled out that the level of detection and level of quantification depends on the product and the maximum daily dose which you take. So that's all cleared up, but it was a moving target during the last 3 years. If we go to the next slide, then you can see the key events towards the current EMA and US FDA guidelines. As I said, it started in 2018 with the valsartan, which was recalled for the presence of NDMA. This valsartan was from a specific supplier, and while looking at the root cause of this NDMA, it was clear that it came from a process where an azide process was used, and there was a formation of NDMA during the route of synthesis. In the same month, in July, EMA then initiated a so-called Article 31 referral, and this and asked to each and every supplier who were supplied valsartan to the market to assess whether there was any risk for formation of NDMA in this specific route of synthesis. Later in September, another nitrosamine was identified in the valsartan, which was called NDEA. And therefore, EMA also extended their referral to look for any other sartan, and all the suppliers were sent this referral and were asked to give their responses by a firm deadline of October 31, which we needed to submit all the risk assessment and confirmatory testing results of the risks. Later on in the year, in December, there was NMPA found in one of the sartan families. And so you can see clearly that the different nitrosamines increased during the timeline. In February 2019, EMA asked for a scientific opinion from the CHMP committee to look at their EMA referral to have backup from scientific point of view to see whether the guidance which was given was scientifically sound. In that same month, also US FDA then came with a formal publication to each and every supplier in so-called FDA General Advice letters. These FDA General Advice letters had couple of points in it. For API suppliers, it was said that even API suppliers needed to issue a field alert report in case they would find any nitrosamine in their API, although normally API suppliers are not forced to issue any field alert report. Also, it was said that all the batches which were on the market needed to be retrospectively tested. And last but not least, their guidance gave that the nitrosamines needed to be undetectable. While in their general advice letters, there were a couple of different analytical methods mentioned with different LOQs and LODs. So it wasn't very clear what was meant by undetectable at that moment. Then a main milestone in 2019 or event was in April 26, where EMA issued the news that in another product, which was not part of the sartan family, pioglitazone, also NDMA was found. And that actually changed this whole story from looking at only the sartan family towards where we are today. So based on this event, also in June, EMA started to draft a non-sartan proposal to assess all the chemical synthesis APIs and drug products to be assessed for nitrosamines, and they formally issued their guidance in September of 2019 with strict deadlines. Also in June 2019, EMA and EDQM issued for the sartans revised monographs and additional interim limits for other nitrosamines. In 2020, some key events in the February, EMA also asked for CHMP opinion for their EMA guidances. And based on the opinion of CHMP, in August of that same year, EMA issued a revised guidance where they also included the bioproducts and the fermentation products as part of all the products which needed to be assessed. Also in that guidance, acceptable intakes were further revised, and some other items were changed compared to their guidance from 2019. And then back in September 2020, also FDA came with a guidance on all other APIs which needed to be assessed. There are some differences in that, and we will go in a little bit more detail in the slides to come. If I go to the next slide, then just a quick overview of the key nitrosamine impurities which are mentioned in the guidelines. There are 7 which are the most known, which are also spelled out with specific acceptable intakes. The chemical name is underneath. Sometimes there are different letters used for the same chemical entity, but it's not limited to those 7. It's clear that for the risk assessment, we need to look at each and every nitrosamine which can be formed. And but those are the 7 main ones. And more information will now be given by my colleague, Lena.
Thank you, Diana. Hi, my name is Lena Ben Mocha Lerman, and I'm heading Lifecycle Management at Teva API R&D. So let's give a closer look how we should assess our APIs for nitrosamines risk. Okay, so the process of assessment consists of 3 main steps. Step 1 is theoretical assessment whether the nitrosamine can be or cannot be in our API. In case we conclude that there is a potential risk, we need to proceed to step 2, which is confirmatory testing. It is important to highlight that during confirmatory testing, we should test nitrosamines which are relevant to our process for a specific API. There is no need to test something that is not relevant for our process. Now, in case during confirmatory testing, we detect levels of nitrosamines which are higher than the limit, we need to make changes in our process. So here we're going to step 3. Now let's deep dive into each and every step. Okay, step 1. When we do theoretical assessment, we should address 3 main sources of potential nitrosamines formation. The first root cause is obviously the chemical process. Then second source can be process water, and the third main source is cross-contamination risk coming from raw materials, solvents, or shared production lines. Okay, so if we touch now deeply chemical process, what should we look into it? Based on organic chemistry principles, nitrosamines can be formed when secondary amine reacts with nitrite ion. But when we assess our APIs, we should pay attention to the fact that we are looking for traces of nitrosamines. So not necessarily we have to use a secondary amine in the process or an ion of sodium nitrate. We can use their potential sources. What do I mean by that? Secondary amine can be found in tertiary amines, quaternary amines. You can find it in some commonly used organic solvents such as NDMA, NMP, and DMF. Also, it is important to emphasize that our API can be a secondary amine itself, and also our API intermediates can be secondary amine themselves. So all those potential sources should be addressed. Similarly, nitrate ion can come from nitric acid, can come from hydroxylamine use. So we need to look at this risk in a very wide way. Okay. Now it is important to mention that obviously the highest risk of formation would be when both components are being used in the same synthetic steps. But it is not a necessity. You can use a secondary amine source in the first step and nitric acid in 4 steps later, and still you might have a risk because of potential carryover. So when we look at our processes, we consider possible carryover, and if there is 2 components, nitric ion source and secondary amine source in our process, regardless how far they are used one from another, we conclude a potential risk, and therefore we proceed to confirmatory testing. Now, with respect to the process water, what kind of risk comes from process water? Process water might contain nitrate ion. So we need to test process water to review our process and to understand the volumes of water that we use in the process. And together with concentration detected, we can conclude if there is risk or if there is no risk. Another possible source for nitrosamines, it can be contaminated raw materials or solvents. So to address this risk, we created the dedicated questionnaires, which we sent to all our vendors. Based on their answers, which were reviewed deeply, we conclude if there is a risk or there is no risk. If there is a question mark, we conclude that there is a potential risk and we proceed to confirmatory testing. And obviously, if there is a risk coming from those questionnaires, we also test our materials eventually. Another potential source is cross-contaminated production line. We assess this parameter as well, addressing the fact that in case the API is produced on a shared line and not on dedicated line, we assess which product was produced there before, and if that particular product might bring nitrosamines risk that can be carried over to other API. Okay, so this is basically all the principles of theoretical assessment. Now, as I have mentioned, in case we conclude a potential risk, we need to take a step 2, which is confirmatory testing. Okay. Both EMA and FDA describe deeply their demands for confirmatory testing in terms of sensitivity of methods, in terms of number of batches that should be tested. We fully follow those guidelines, and we develop and validate sensitive analytical methods using sophisticated equipment such as LCMS, triple quad, GCMS, triple quad. Now I hand over to my colleague Vesna.
Thank you, Lena. Hello to everyone. I'm Vesna Prgomet, Director in Teva API Global Regulatory Affairs. So now let's move to step 3, actually making a change after all the assessments and everything we have done. As Lena explained, after we have concluded and all risk assessment activities in step 1 and have done confirmatory testing where needed as step 2, it is time for step 3, making the required changes in order to control nitrosamines. Following changes may be required. One is change in manufacturing process. For example, it can be additional purification step, some minor process adjustment, or even adding a process step. Then we need to introduce appropriate specifications. As mentioned before, methods are already developed and validated at stage 2. Besides changes in process, measures related to the premises and equipment could be needed, like cleaning procedures, environmental monitoring, etc. And last important thing is to control any future change that may impact on this risk. It can be change in a supplier, change of manufacturing process, change of packaging. So any change that could have an impact needs to be evaluated for risk nitrosamines in the API. When we have done all what is necessary to implement the change, it is time to communicate it to our customers, MA holders, and authorities. And finally, it comes to a submission of a change to regulatory authorities. As API manufacturer and CP holder, we submit CP revision to EDQM. Depending on a time and the extent of change in process, it could be notification, minor or major revision. Even it can end up as a sister CP. Inclusion of mutagenic impurity in API specification is considered minor revision by EDQM. Any update in impurity sections is also minor revision, even if it's only risk assessment concluding that there is no risk is being added. EDQM requires this also to be submitted as a minor revision, which is then reviewed and approved. Once updated CP is approved, it should be filed by MA holders through either type 1A or 1B variation as applicable. All CPs should be revised in order to control nitrosamines by September 2022 at latest. Now coming to responsibility of MA holders. Final responsibility is on them, meaning that the drug product needs to be in line with recommended limits. Following EMA guidance, MA holders have responsibility of submitting appropriate variation for changes in active substance master file or DMFs. Therefore, we continuously communicate outcomes of each step to our customer, and if there is a need, we take care that updated DMF is ready to be submitted by DMF holder, which is us, Teva, upon request. Depending on the extent, change in process can be classified as type 1A, 1B, or even type 2 according to variation guidance. Any change in specification, which includes addition of nitrosamines impurity, is classified as type 1B. Deadline for MA holders to report changes to EMA is September 26 in 2022, and we aim for timely support. In case API is covered by CEP, update CEP should be submitted by MA holder through type 1A or 1B variation as applicable according to variation guidance. And for US market, DMF amendment that includes relevant updates should be prepared and submitted to FDA in collaboration with MA holders, or for US market, it's usually called ANDA holders. So MA holders or ANDA holders are notified in advance when DMF update will be ready, and we work together to be ready on time. Deadline for MA holders to submit changes to FDA is October 1st, 2023. FDA allows proposed process to be submitted with estimate for removal of the original process in the same DMF. The different synthetic processes should be identified by separate codes. Only in case if the original process cannot be discontinued within a reasonable time frame, the new or revised process should be submitted in separate DMF. Change category is defined following relevant guidelines depending on a change extent. And also, it should be discussed with MA holders based on the extent and type of the change. With all this said, we are moving to summary slide and back to Lena.
Okay, so this is the summary slide summarizing timelines for each and every step, and few details about sensitivity of methods as demanded by European and United States authorities. So step 1, which is the theoretical assessment, based on both authorities, should be ready by March 2021. Confirmatory testing and change of the process if needed. Have a deadline for United States till September 2023 and for Europe till September 2022. Now, what about sensitivity of methods? Emma has clearly described that QA LOQ should be less than 10% of the limit. If we have levels higher than 10% of acceptable intake, we should put this nitrosamine in our routine release. As for FDA, they are more referring connecting sensitivity of method to dosage form. For products having maximum daily dose less than 880 milligram per day, LOQ should be around 30 ppbs, and for higher dosage form, it will be even less. That's it.
Thanks so much, Diana, Lena, and Vesna. It was very, very interesting. We received quite a few questions while you were all speaking. So we're going to go through some of them now, see how many we have time for. So the first one is, how many products have been reviewed so far?
Okay, I think that I will take this one. So we have reviewed all our commercial products as well as pipeline products to be launched. We embedded this methodology of nitrosamine assessment in all our new products that are under development at our R&D center. So it will be an integral part of our assessment of mutagenic impurities from now on.
Great, thank you. Next question is, how do you treat AI for nitrosamines not listed in the guidelines, for example, generated from an API or from process intermediaries?
Shall I take that one? Yeah, it's a pretty good question. The guidelines give a full table for those which are known. What we do if the nitrosamines are not known, we look into the information to see whether there is any lookalike molecule already mentioned and measured in toxicological studies. And if that's not the case, then you should take the most conservative approach as mentioned in the IC 877. So I hope that answers the question.
Thanks a lot. Thank you. Next question is, when and how are you informing customers?
Thanks. I think this one is for me. Customers or our MA holders, as I said, are informed after each step. So after we concluded step 1, risk assessment, we have shared statements with information and conclusion if there is a risk or there is not a risk. If yes, then we have estimated by when confirmatory testing should be conducted and next information will be shared. Then, depending on an outcome of confirmatory testing, there could be need for making a change or not. Again, we are notifying our customers with relevant information and timelines for change implementation or readiness to be submitted at their site.
Thank you. We have time for a couple more. So how do you define permitted levels of nitrosamines in APIs?
I will take this one. So limit for nitrosamines in API is calculated by dividing acceptable intake for specific nitrosamine by maximum daily dose of the API. This is how we establish the limit.
Okay, and the last question I think we'll take now is, how do you establish permitted levels of nitrosamines if more than one nitrosamine is present?
Yeah, I can answer this one. If there is more than one nitrosamine present, we need to assume that the most potent one is the limiting factor. And we need to assume that all the nitrosamines need to be summed up to those limits and cannot be beyond that limit of the most potent one. So you take them all together, you take the sum, and then you take the most stringent limit from the most potent one. That's how we deal with it.
Great, thank you very much. Thank you. So that's about all we have time for today. Thank you so much to our speakers and to our lovely audience for joining us. Don't worry, if we didn't manage to answer your question, we will try to reach out personally to you afterwards to answer it. Thank you for joining us, and we'll see you again soon.